Well, picking up where we left off here in Chapter 8, we were talking about some of the milestones that we know can occur in the very earliest stages of brain development, mostly prenatally, as we looked at the stages taking us from neurogenesis to neuromigration and specialization and cell differentiation and ultimately arborization, right, the formation of synapses. But something else that we want to also think about when it comes to looking at development is the relationship between brain development and the emergence of skills and abilities in infancy and early childhood especially, but of course this would span easily through middle childhood, adolescence, and even a little bit of early adulthood as well. And even further on that topic, as we think about aging, we could also take these three perspectives that are listed here and understand how certain changes in the brain relate to some age-related declines that we may experience. And I think we talked a little bit about some age-related changes in memory in the first half of this chapter recording here. But let's first take a look at these three different kind of approaches for how to study the relationship between brain development and the emergence of various behaviors. And then we'll have a few examples to round this out. And hopefully we'll get this chapter done fairly quickly here. So the first one listed here is to watch for structural development, right? That is to look for changes in the brain, right? Watch and see what kind of brain changes are happening. And then once we observe those changes in the brain, we can then find certain developmental milestones that are occurring behaviorally that we could tie closely to those brain changes, right? So the very first thing is you notice that the brain has changed because you're monitoring some specific brain structure by perhaps doing what's called a longitudinal study. Longitudinal studies involve tracking development over a period of time within a given individual or group of individuals. So maybe you're doing some brain scan for some period of time, and you're watching some specific part of the brain and seeing how it changes over time. Now, how might it change? Well, it can grow, it can get bigger, right? We can also find that it can show some increase in connectivity. So we're talking about maybe synaptogenesis, and we're seeing some increasing arborization occurring in a certain region of the brain, and that might show up as maybe a thickening of the cortical layers. Conversely, we might also see a pruning occur, and therefore that might actually manifest as a thinning of some of the cortical layers, because what's happening there is that unnecessary connections are being pruned away, which, as we have also previously covered, suggests that the brain is increasing its efficiency, that unnecessary synapses and therefore things that might be a net drain on brain metabolic function can be pruned away, and that improves overall efficiency, as well as myelination occurring, right? So we start to see neurons in various aspects of the cortex myelinate, and that's going to be associated with the speeding up of processing of information in that part of the brain as well, right? So if we observe these particular structural changes, then we can also then administer some sort of psychological, behavioral, cognitive, perceptual test to the infant or child and see what sort of changes are happening behaviorally as we are watching those changes in the brain occur over some period of time. So just as one example here, there's some research that's observing in early childhood how frontal lobe connectivity begins to increase during certain stages in early childhood. And as that frontal lobe connectivity is increasing, some of the core milestones that we will see would be the emergence of certain social skills. So social skills could involve things like empathy, right? Or theory of mind, the ability to understand the mental states of others, for example. A classic theory of mind test is known as the Sally-Ann test. And you may have encountered this maybe in a developmental psych class. The Sally-Ann test is this idea of a story that centers around two characters, Sally and Anne, and they're playing with a toy in a room. And Sally takes the toy and she hides it in some location, let's say in a box. And then she leaves and she goes outside. And while she's outside, Anne takes the toy and moves it from a box to a basket. And now when Sally comes back into the room and she wants to get her toy back, where is she going to look for it? And the right answer, obviously, is she's going to look in the box because that's where she left it. But young children under the age of 4 typically will say that she's going to look in the basket because they fail to have the ability to understand that Sally's knowledge and access to information is not the same as the child's knowledge, which the child knows that Anne has moved the toy. That was part of the story. But separating what we know about something having happened versus what some other person may or may not know is a form of a social skill that we have, a social cognitive ability to understand that other people's knowledge may be different from our own, and to have that boundary between our own subjective knowledge and others. And of course, that's an important part of social behavior, to be able to sort of take another person's perspective and understand how other people understand and think about the world, as well as, of course, feel, which was, I mentioned, empathy. So if we can see that the brain is there are certain frontal lobe changes that are occurring as we see the emergence of empathy and theory of mind type capabilities, then certainly we could draw some connection there between those two things, right? Here's another way to think about how to do this research, though. What if we're doing it maybe the converse way, right? What if we are watching a child's behavior and we are noting that there are certain changes that are starting to happen? In this case, we maybe talk about language, right? So we might say, oh yeah, we can see the child has, here's another language milestone. Child gets their first word when they're about 1 year of age. When a child is 2 years of age, they may have as many as 1,000 words. That would be pretty precocious, but maybe more commonly as an average would be 700 words on their second birthday. But that's a lot, right? To go from having 1 word in your vocabulary at the age of 12 months to having at least 700 words in your vocabulary by the time you're 24 months, right? That's a lot of language learning, at least word learning, that occurs in the span of a single year. So that's a lot. And we might think that, well, there must be something in the brain that is changing to support that. And we already know from other research that we've talked about, like the autopsy studies from Wernicke, to give us Wernicke's area and Broca's area. So we could now generate a hypothesis that says that now that I'm observing this child showing these language milestones from age 1 to age 2, maybe I could make a prediction that there should be a corresponding change in one of those language parts of the brain. I think specifically we might look towards Wernicke's area to see that maybe there's some increasing connectivity there, some increasing myelination, something just must be happening in that part of the brain that mirrors that all these language milestones that are occurring between the first and second birthday. Another thing that we could do is we could look at external other third factors in a way, right? So here's an example where, what if there is a brain injury? And so in the case of that, we might find then that that injury affects brain structure, but it might also then result in the delay of certain milestones. This can happen, for example, certainly unfortunate to talk about, but oftentimes in birth, a child may experience some loss of oxygen, right? So this is called anoxia or hypoxia, right? So either total loss of oxygen or reduced oxygen. And this can sometimes damage certain parts of the brain. And it's known, for example, to sometimes result in epilepsy or cerebral palsy. There can be various things that occur from a hypoxic state like that. And so we might see that there could be some brain injury that results from this. And now, once we understand where, maybe from an imaging study, where the brain injury occurred, we can observe how there are certain maybe delays in achieving certain developmental milestones as a result of that. Could be language-related milestones, could be something else, right? But the idea is that we can map these relationships together. Here's an additional make PowerPoint work here, example here. So in this case, we see over the span of the first year of life, there are certain milestones that are associated with reaching and grasping, right? By the time children are about 2 months of age, they have a very crude ability to kind of swipe their hands around and reach for objects that are within reach. But they really kind of do a whole hand grasping of it. They're not very accurate with reaching and picking it up with their fingers. But a couple months later, they get better at grasping objects. And what we're suggesting here with this box is that they can sort of shape their grip to match the shape of an object. And so they're getting better at kind of understanding how to appropriately use your fingers to wrap around objects of different shapes and sizes, right? And that's something that's important when you're grasping something, right? Is you need to tailor the nature of your gripping behavior to the orientation of an object and its size and its shape, because you want to be able to grasp it in an optimal way so that you don't drop it, for example. And we see even further advances here. By the time a child is 10 months of age, they're capable of doing something called a pincer grasp. And so that means actually using your thumb and forefinger to pick up small objects. So that requires a great deal of fine motor skill and motor precision to do that. So this is an example, again, of kind of like the second point on the previous slide, that we're going to observe a series of developmental milestones. And then we would think, well, what's going on in the brain to support this, right? We kind of know where to look. We would think to look in the motor cortex, perhaps in the frontal lobe, but there may be other things happening with the basal ganglia and cerebellum that could also be involved here. And so we should be able to expect to say that before each one of these milestones, there should be some corresponding developmental process in the brain. And we could maybe go in there. And once we know when these milestones occur, that then serves as a guideline, right? So the question here is not so much that we would not be surprised to find the motor cortex changing, but the idea is that now that we know that these milestones occur at very specific times because of decades of research on infants, that tells us, yes, we can expect at 2 months, we're going to see this big kind of reaching behavior, and at 4 months, we're going to see this other kind of reaching behavior. Then these known timelines for things can guide the neuroscience research to say, okay, now is a good time that we have a group of babies that are 4 months of age, or maybe just a little bit shy of 4 months. Let's go in and start looking at the motor cortex right now and see what's happening just as we are moving into this ability to start grasping more accurately at 4 months. Or then we wait until they're almost 10 months of age and we start studying. Now that we expect that pincer grasp is going to emerge any day now, let's see what's happening in these children's brains as they gear up towards that skill. And what we find in this case is oftentimes some combination of some of the things I was listing earlier: increased myelination of neurons in the motor cortex of the brain, sometimes also a pruning of some extraneous connections in the motor areas of the brain. And these two things together generally speed up overall brain efficiency and the capacity to process information. And so clearly there's some degree of information processing in the brain that's required to support this kind of advanced skill of pincer grasping. Speaking of language again, here's another example. So in this case, what we would again suggest is that once we know certain language milestones, like I said before, we're going to start with clear signs of language use by the time we're 1 year of age with the first word. Now there's some stuff before then too. So talking about how children kind of learn how to babble and, go goo goo gaga kind of stuff, right? But we'll say that, we're going to get that first word at 1 year of age. And in that second year of life, leading up to the second birthday, there's going to be many hundred words acquired. And likewise, there's going to be more than just word learning, right? There's also a lot of grammatical stuff that has to occur. And grammar is really the rules of language. How do we put words together to start forming sentences? And there's a certain developmental path that we understand there, that children go through these simple constructions known as telegraphic speech that are very simplified, short 2 to 3 word sentences, until they start producing more grammatically fluent language when they're 4 and 5 years of age. And even still, there would be continual improvements in language ability all the way up until age 12. So again, what we can do is we can track how the brain is changing in those language centers. Again, we'll look at Brokaw's area, especially in this case when we're talking about a grammatical production, right? So when it comes to word learning, we might want to understand what's happening in Wernicke's area. But if we want to look at the development of grammatical language, right, the ability to produce complex grammatical statements and novel statements, we would want to look at Brokaw's area for that and understand how that part of the brain is changing and how that mirrors and correlates to these changes in linguistic sophistication in childhood, right? And again, as I said, there's going to be a lot of these kinds of findings here. Increased arborization. Here it's written as dendritic complexity, so just meaning more dendrites, right? More branches there, more synapses, and also increasing the myelination of these as well. It's worth also mentioning that we'd find some myelination in the motor cortex for this as well, because clearly when it comes to producing language, Brokaw's area is involved in kind of organizing the grammatical structure of the language, but we still also need to use the motor cortex to initiate the actual movements involved in speech. That is the movement of the vocal tract, the lips, tongue, and jaw. We have to perform a lot of complex, precise movements with those speech articulators in order to be able to produce language. And so we would expect to see these changes occurring not just in Wernicke's area or in Brokaw's area, but also the motor areas of the brain that specifically control those particular moving parts. Let's think about this one as well. So here's another example. Here we're going to appeal to some other known milestones in development, this time cognitive development. So Piaget gives us these fairly well-known 4 stages of cognitive development that occur in childhood. So the first couple of years of life, more or less, is known as the sensory motor stage. And the idea here is that children are just learning how to understand the world through their senses, right? Their visual system is developing, their sense of touch is developing, and at the same time that those sensory and perceptual systems are developing, their motor skills are developing, and they're learning about certain properties of objects in the world and how to move around and interact with these objects, right? So this is the basic idea for that particular stage. And then once we move into the early childhood, toddler years, children now move into what he calls pre-operational thinking. And this means that they're starting to think symbolically. They can generate mental representations. And of course, now the emergence of language and words, which are symbols that stand for things, are all evidence of this kind of symbolic thinking. But Piaget refers to it as pre-operational thinking because of the idea here that one of the things that Piaget is really focused on is operational thinking, which is logical thinking. And children at this stage are infamously incapable of good logical thinking. They fall victim to a variety of logical errors that Piaget devised to test for. One of the more famous ones is called the conservation task, right? And this is a famous thing. You've probably seen this before, where if you take 2 glasses of water that are identical in shape and size, and you put the same amount of liquid in both of them, and put those in front of a 3 or 4-year-old child, they can correctly say that they have the same amount of liquid in each glass. But if you take one of those glasses and you pour it into a taller, skinnier glass, now that water is going to change its level, and you take this new glass with a higher level of water, and you put it with one of the other glasses that still has the original amount, and you say, now, do these still have the same amount, or does one have more? And they're going to change their answer and say that this new glass has more, even though they saw you pour that fixed amount of water into it. Because it's changed its appearance, they are not able to take into account that change in appearance as also a change in the shape and size and dimensions of the glass. So now they start getting it wrong, right? They just don't understand the concept of conservation of volume. They don't understand how it's a reversible event. So they just kind of get fooled by this change in appearance. But once we move into, approximately the school-aged years here, they start being capable of logical thought. This is called concrete operational thinking. And then moving into adolescence, we become more capable of complex logic, abstract thinking, and that's what Piaget calls formal operational thinking. And this would be seen as the culmination of human logical thought, at least as far as Piaget is concerned here, right? And so we might think that, all right, if Piaget is correct here, and that development really proceeds in terms of these 4 stages, and that it occurs around these same times that we're expecting it here, so somewhere around the 2nd birthday, the 6th or 7th birthday, and the 12th birthday, give or take or so, then we would expect to see certain brain changes coincide with those very important transitional ages, right? And this is where we see Epstein's research coming in here, where he found that there were in fact, certain rapid changes in brain development and brain complexity that roughly correspond to the proposed ages where you transition here. Although, as noted here, there's a 5th one. So Piaget only has 4 transitional points, right? From stage 1 to stage 2, stage 2 to stage 3, stage 3 to stage 4. But there were extra ones. So there was, I should say onset. So there's one at birth as well. So I left that one out. So that's 4. An extra one at the middle teenage years, 14 to 16 years. So that's a provocative finding, right? Because one of the things that this could do is it might cause us to revisit Piaget's theory and ask whether or not there shouldn't be a 5th stage here, right? Are there some other additional cognitive changes that happen in that 14 to 16 year transitional area that could explain some new skill that emerges beyond what was acquired at around age 12 or 13, right? And so maybe there's some extra stage, and that would be an interesting possibility to explore scientifically, is to think that, well, Piaget proposed these 4 stages. We can identify some approximate neural changes that correspond to these, but surprise, we've identified a 5th neural change occurring a little bit later on. So does that mean that we should explore the possibility of an extra stage, that we might go back and revise Piaget's work here? So far, that really hasn't panned out, but the idea is that's the kind of discovery that could open up new avenues of research here. Something else very important that we need to understand about brain development relates to one of the things that I had covered in the first half of this recording here, looking at the importance of experience here, right? So we said that a lot of brain development may be genetically regulated, especially the more simple parts of the brain. But what about the cortex, the neocortex? This is where all the action is for higher cognition. This is the part that is much larger in humans than it is in other animals. It seems to explain why humans have this ability for the flexible behaviors that we talked about in a previous chapter, compared to all of the fixed behaviors that we may find in animals, right? And non-human animals, I mean. So this ability of the brain to change, which we had called plasticity at the beginning of this chapter, appears to be very, very important in humans, especially because that's where we get flexibility, that's where we get learning. But we need to understand exactly how the brain changes. How does the brain learn? How does it adapt? What kinds of changes does it undergo? There are a few later chapters where we're going to really get into some detail about how memories get encoded in the brain with a concept known as long-term potentiation. But before we get there, we can sort of start with a very simple idea known as Hebb's law. So this research done by Donald Hebb in the late 1940s suggests something here. And so what Hebb had noted was that when you look at animals that are raised in, let me back up. He's studying brain development in young animals, and is one of the ones who initially discovered this idea of synaptic pruning, right? The fact that unnecessary and unused synapses in the brain are just pruned away, or sometimes, of course, unused neurons kill themselves off through apoptosis, right? So Hebb develops a principle to explain how it is the case that when a synapse gets used, that helps these neurons stay alive. And if a synapse is not getting used, that results in the pruning away of these synapses and sometimes of entire neurons. And this is called Hebb's law. And so the simple way to describe it is this little quote right here: neurons that fire together, wire together. And what that means, the firing together just means the activation of a synapse, right? And the action potential is the firing we're talking about here. Now, what does it mean for the neurons to wire together? There's a term sometimes used in neuroscience, and this kind of relates to the concept of the electrical circuit metaphor that we sometimes use to talk about neural networks, is that they're wired together, right? But you can talk about something like a hardwired system, right? And a hardwired system is when the circuits are really closely tied together. And that's the argument here, is that something happens that when a particular synapse is repeatedly activated, that makes this connection become really strong. Now, there's another quote down here in slightly smaller text that's really a direct quote from Hebb's. So the neurons that fire together, wire together is really just sort of a kind of a simple little phrase that captures the essence of the idea. But here's exactly what Hebb wrote. When the axon of a cell A is close enough to excite a B cell and takes part on its activation in a repetitive and persistent way, some type of growth process or metabolic change takes place in one or both cells so that increases the efficiency of cell A in the activation of B, right? So that's a long sentence to get to that point. But what it's really saying here, we should remember the nature of synapses. We talked about ionotropic synapses, but I also talked about metabotropic synapses, right? And the second messenger signal inside of a neuron that might do things like cause additional ion channels to be built into the cell membrane. And so that's what this specifically is referring to when it says that when neuron A repeatedly activates neuron B, some type of growth process or metabolic change takes place in one or both cells. So if cell B is the postsynaptic neuron, maybe it's going to grow more dendrites or dendritic spines. And then within new dendritic spines, it's going to grow more receptors, more ion channels, right? Likewise, neuron A might also, that's the presynaptic neuron, might experience metabolic changes that cause its axons to branch out a little bit further to stimulate these extra dendritic spines that form on the postsynaptic neuron. And then correspondingly, neuron A may need to manufacture more neurotransmitter to support all of these extra connections between these two cells now. And so now you've gone from maybe a very small connection, just a few axon terminals and a few dendritic spines kind of touching each other, to now that sort of spreads out to be a much more arbored, if you want to use that as a verb there, or I guess adjective, a connection between those two cells, right? So the dendrites branch out a little bit more, the axon branches out too, and now you have all of these interconnections. And so clearly now the synapse between these two neurons is much stronger than it was before. It's a lot easier for A to repeatedly stimulate neuron B now. The opposite would occur if there is never any activation of this synapse. Then there would be metabolic changes that cause these dendrites or axons to actually shrink away, and they get pruned out, right? Now, evidence of this comes from a lot of studies where they looked at research. So Donald Hebb was a big pioneer in this, as well as Harry Harlow and a few others back in the 1940s and 50s looking at this, where they wanted to look at animals that are raised in these controlled environments. So they want to say, okay, the idea here is that in order to stimulate a synapse, you have to process a certain kind of piece of information that needs that synapse, right? So let's take an example here about language, right? There's certain parts of the brain that we use for processing language when we hear it, right? And that happens a lot in Wernicke's area. So if we are exposed to language in infancy and early childhood, then all of the synapses that are part of that processing of those sounds of language are going to be repeatedly activated. And that's going to strengthen those synapses, and they're going to get stronger and stronger and stronger. And that's going to make us even better at processing the sounds of the language that we are exposed to. But what if we're not exposed to any language, right? That means you're going to have a bunch of synapses because, again, synaptogenesis just happens in the brain, but you're not going to be exposed to it. And if you're not exposed to any language, all of these synapses that are there waiting for linguistic input never get activated. And so after some period of time goes by, if nothing is happening to activate these synapses because you're not being exposed to any language, then you're going to end up pruning those synapses away. And that lack of exposure, this deprived environment that deprives this one unfortunate child now of any exposure to language results in really a shrinking of that appropriate brain area and an atrophy of that brain area, because now you're going to start a lot of apoptosis and synaptic pruning, and you're going to have much less neural connectivity in that region of the brain as a result of this lack of exposure, right? And so it's that kind of thinking that led to Hebb and others to do this kind of more controlled research with animals to say, okay, what happens when we raise an animal in a rich environment that has lots of interesting things to see and do and hear, and that's going to activate more synapses. But if we keep them in a deprived environment, then there are fewer things for them to see, do, and hear. So that's going to activate fewer synapses, and we might expect there to as a result, for there to be less connectivity in the brain, right? Smaller brains with fewer synapses and things of that nature. By the way, back to that language example real quick, there's one additional point that's worth pointing out about this exuberant synaptogenesis. There is no way for your brain to know in advance what language you're going to be exposed to, right? You might be exposed to English, you might be exposed to Spanish or German or Italian or whatever it might be, right? So the idea is that there's any number of languages, hundreds of languages that you could be exposed to. And what's very important is that languages don't just differ when it comes to words and a little bit of grammatical rules, but they oftentimes differ in terms of the actual sounds used, what are called the phonemes, the building blocks of language. And some languages just really sound different than others as a result of this, right? And since your brain has no way to know whether you're going to be exposed to one language or another before you're born and before you actually start hearing the language that your parents are speaking, the brain overproduces the synapses, right? It produces enough synapses so that way it doesn't matter what sound you're exposed to and what language you're exposed to, the brain will be ready to go to be processing all of those sounds, right? But what's likewise going to happen is that in the course of that first year of life, in most cases, children may be exposed to just one language, or maybe two if they're in a bilingual home, but rarely more than that. And so now you're going to end up keeping, as a result of Hebb's law, you're going to end up keeping the synapses that are useful for processing the sounds of the language that you're exposed to. But all of the sounds that could be part of some other language and you're just never exposed to it in infancy, those will get pruned away, right? And that's partly what makes it more difficult to acquire a second language or a third language or whatever it might be later on in life, because the plasticity of the brain in early childhood is very much a function of having synapses there to process those experiences if they were to happen. But if those experiences never happen, then they end up getting pruned away, and that reduces the overall plasticity of the brain and makes it much more difficult to later acquire those particular skills. Here, by the way, is an example of the kind of rat experiment we're talking about. So this is an example of a particularly enriched rat environment, right? It has many layers and levels with ramps that go up and down and various objects for them to interact with and climb on and so forth. And so there are lots of things to see and do for these rats. And that's really different from a rat kept in a standard laboratory cage, right? Which would just be a very bare bones, simple cage with has nothing but a little food tray and a little water bottle to drink out of. And so here's just an example of the kind of arborization of neurons in their brains that you would see. And so clearly the ones that are kept in the enriched environment just have many more dendrites and more complex connections, increased synapses. And as the previous slide pointed out, also not just more synapses, but more astrocytes. And that's very interesting because remember the astrocytes are part of the blood-brain barrier and they feed the neurons. And so, of course, that means that these neurons are going to get more nourishment because, of course, they're being used and we would have more astrocytes as well. Here's something else that gives us another view of what's happening here. So we're going to see in a coming chapter that in the visual cortex, there are different columns in the visual cortex that process information from the left eye versus the right eye. And the organization of visual input into these two separate visual columns helps not just keep the left eye, right eye signal separate, but actually it's also important for integrating the two of them because we use two eyes to achieve binocular depth perception, right? And what we're seeing here is an example of looking at kittens. And what we want to do is we want to study the arborization of neurons in different columns of the visual cortex that process left eye and right eye. And what they did in one of these experiments is they would keep one eye closed. They would essentially just sew it shut during their early development when they would normally open their eyes for the first time. But here they kept it sewn shut for a week during a very critical time in early visual exposure. In this case, it's the right eye. And what we see here is that in very early infancy, there's not really any noticeable changes, but starting in adolescence, we start to see some detectable changes in the arborization of the right eye neurons. And then what we find is that in further adulthood development, these neurons really fail to fully properly develop, and there is significantly reduced dendritic complexity in these neurons, right? So we see then that would be more of a long-lasting, even permanent deficit in their visual ability. Their right eye's ability to process information would be very weak compared to their left eye. And likewise, their ability to integrate the two eyes to get binocular depth information would also be significantly impaired. Now, we clearly don't do this kind of research on humans, right? These kinds of, let's take a human infant and intentionally raise them in some deprived environment or do something to prevent visual input or language exposure. Well, those would be highly unethical. Even the animal research, of course, was of questionable ethics. And at the time, it may have been more possible to do those studies in the 1940s and 50s than it would be today even to do that, because there have been changes in ethical standards for research even on animals. But what we can do is we can study other situations in which children might be kept, like, I mentioned the issue earlier of language and what happens if you're not exposed to language. And while we would never do that intentionally, we do know of a few case studies, especially the infamous story of Jeannie. And you may have heard of Jeannie in other courses as well. And so I don't really want to go into all the details there. But the idea is that she was raised without any exposure to language, as best as we can tell, all the way up until the age of 13. And then later showed that it was essentially impossible to teach her how to speak and how to really have a good any kind of real grammatical ability to speak language. And so what we know is that when children are deprived in those kinds of cases, it can be very damaging to their development. But what we also know is that it also depends on the duration of the exposure, right? So in the case of Jeannie, because that duration was basically her entire childhood up until the age of 13, she had very severe deficits. But in other cases, if deprivation is shorter, then the brain may have the ability to actually overcome some of that. And because the human brain is so plastic and so adaptable compared to other animals, some of the stuff that we see, just like with this kitten research, may not fully generalize to humans, because the human brain maybe has an even stronger ability to overcome and adapt. But careful, controlled research would never be done to really make that direct comparison. Some other things worth pointing out here is that other things can injure the brain. So we were talking about injuries like anoxia earlier at birth, but stress is also something, right? So early childhood stressful experiences could alter brain development. What we know, for example, is that children who are chronically stressed and activates the fight or flight response, it repeatedly activates the amygdala. And what you might think would occur does occur, which is that based on Hebb's law, repeated stimulation of neurons in the amygdala result in increased arborization of the amygdala. So now the amygdala itself becomes, as a result of Hebb's law, easier to activate in adulthood. So, that's going to have some implications, right, for adulthood. How does a child who has gone through these highly stressful experiences that has maybe sensitized their amygdala, which activates in fear and aggression type behaviors, what's their emotional response going to be like later in adolescence and adulthood? Will it be altered? Well, that we might be able to make some predictions about that. We also know that stress hormones, not only do they activate the amygdala, but they usually inhibit the hippocampus, right? Cortisol does some stuff like this. And the hippocampus, of course, is important for embedding and encoding new long-term memories. So not only is this child possibly sensitized to these negative emotions, but they're going to have impaired memory potentially as well. So we can see that there's a lot of research on the negative health effects of stress, but a lot of times this focuses on adulthood and how stress can cause a lot of cardiovascular and immune system issues. But here we're looking at early childhood stress can have very profound effects on the development of the emotional centers of the brain and the memory centers of the brain, and maybe even increase the risk of later diagnosis of a mood disorder, depression disorder, anxiety, and so forth. Right. The amygdala and anxiety, of course, are very closely linked with each other. We know that anti-anxiety medications oftentimes work by inhibiting activity in the amygdala. So in particular, we would think that this early exposure could likely increase the likelihood of being diagnosed with an anxiety disorder later on down the line. Other kind of things might also go wrong here. I think this is our last slide, by the way, so let's get this one done. At the very beginning of these notes, we were talking about neurogenesis, neuromigration, and then complexity and arborization. There are some situations where it is known to occur where the neuronal migration does not go exactly according to plan. Once neurons get into place and they start to differentiate into different kinds of neurons, that doesn't always go exactly according to plan. And what this might mean is that different regions of the brain have either too many or too few of a certain type of neuron that needs to be there, or maybe a certain type of glial cell that needs to be there. And that might mean that certain parts of the brain end up either over-connected and have too many synapses, or they're under-connected with too few synapses, right? Some examples here are that certain neurons called pyramidal neurons in the hippocampus have kind of an altered connectivity, and this is known to occur in schizophrenia. We also know that there can be an overproliferation of synapses in some parts of the brain with autism spectrum disorder. Some of these could be in the frontal lobe, some of them could be in the parietal lobe. But the idea is that what we oftentimes identify as a sensory processing disorder in autism spectrum disorder could be associated with overconnected regions that process certain stimuli. And that's why this might also vary from person to person. A lot of times people with sensory processing disorders have very unique issues there. And that may depend on just what specific region of their brain ended up being perhaps overconnected with an overproliferation of synapses. Where sometimes it's the pruning part that doesn't work quite right, right? So you have all these extra synapses and they should get pruned away, but maybe they never get pruned away correctly. So then you keep this kind of over-connected brain. So that's what I actually have in the next slide, is that sometimes you just end up with insufficient pruning, which results in an over-connected brain. Of course, there can be also genetic issues. So there are a number of genetic issues that can affect brain development and result in some atypical form of brain development. So I have listed here Down syndrome, which is a genetic issue. There are a variety of other genetic issues. There can also be teratogenic exposures. So medications, I had briefly mentioned how certain medications might cause neural tube defects earlier, but others can just cause abnormal brain development, right? So alcohol exposure, for example, can result in fetal alcohol syndrome, which affects higher cognition and motor skills and motor coordination. Sometimes a virus like rubella can affect brain development. And then lastly, I'm mentioning something that I already mentioned during this presentation already, which is reduced oxygen at birth. Either hypoxia or anoxia can injure the brain, resulting in sometimes epilepsy, sometimes cerebral palsy, sometimes things that are more severe than either of those two. And so again, though, we can understand where the injury occurred. We can identify, which part of the brain was really deprived of oxygen. And then we can understand, okay, now that we know the injury occurred in this location, let's monitor this child and see where are these delays and deficits going to be. That teaches us some things about brain development and behavior. And of course, even also ask the question about plasticity, because in many cases, a child can overcome and adapt, because the human brain in particular, as I said, does have this amazing ability for plasticity. So understanding how the brain adapts and overcomes from these early injuries is also a very useful thing to be able to do. So anyway, I think that wraps up our chapter here. And so we're done with that one. And then we move on next up with the chapter on vision.